Hello, today we are going to have a new topic and a new chapter of Neoplasia, and our first topic is the Neoplasia. What is Neoplasia? A Neoplasia is an uncontrolled monoclonal proliferation of cells. What is monoclonal? The monoclonal produced by being or composite cells derived from single cells. A single cell proliferate into different cells in abnormal, uncontrolled growth is neoplasia and can be benign or malignant. A neoplastic growth have two components. This thing you have to remember the two components of neoplasia. One is parenchyma and second supporting stroma. What is a parenchyma? The parenchyma is a functional tissue of organ distinguished from the connective supporting tissues. And the parenchyma, they mean neoplastic cells. Basically, parenchyma is a neoplastic cells, and second part is supporting stroma. The supporting stroma are the non-neoplastic cells, and non-neoplastic contains the blood vessels and connective tissues. If you guys see, the book is giving it Venn diagram regarding an normal cells and normal structure. It is having basement membrane with apical pleurectin, means proper sequence, a proper arrangement of cells, and having the normal nucleus and cytoplasm relation. The nucleus having normal size, cytoplasm having normal size. This is the things, basic thing a normal cell having, a normal tissue having, a normal basement membrane, a good sequence, discipline, and a normal size of nucleus and cytoplasm mean a normal nucleus and cytoplasmic connection. Our second thing, the normal cells facing any stimulus. If there's any stimulus that is affecting the normal cell, then it will convert it into a dysplasia. The dysplasia, the loss of uniformity in the cell size and shape. If we talk about a normal cell having a basement membrane and apical polarity, already I told that having a normal sequence and order and discipline, and they are giving us a vital example, we're showing the normal and dysplasia spectrum. The first thing, dysplasia. The dysplasia is loss of uniformity. The uniformity you are showing in normal cells here. It will be disintegrated and it will be lost. The cell size will be increases and the shape is also increases, and it is called pleomorphism. The pleomorphism in multiple forms of morphology within the same species and the same cell having. For example, it is a one cell and it will after dysplasia is looking like that, looking like that, looking like that. The cell is seen, but after a different condition, it is taking different shapes. It is pleomorphism, in which is a lot of tissue orientation and nuclear changes. What I told you, the normal cells is the normal nuclear and cytoplasmic ratio. But in dysplasia, the nuclear and cytoplasmic ratio will be increased. If you get taken the side of nucleus is so much big, fitted in cytoplasm. And it's often reversible. Dysplasia is often reversible, but if we are not controlling that growth, if we are not controlling the dysplasia, then it will convert into carcinoma in situ. It's a pre-invasive stage carcinoma in situ on the surface of the epithelium. And it's an irreversible sphere dysplasia. It is irreversible sphere dysplasia, but it involves entire thickness of epithelium, but does not penetrate the dead basement membrane. The basement membrane will be normal. The basement membrane will be normal. If you guys check, the basement membrane is normal. It is not harming the basement membrane. But till that, carcinoma in situ, all the thickness of epithelium is affected, but the basement membrane is normal. If you are not treating then it will be invasive carcinoma. Invasive carcinoma, the cell have invaded basement membrane. When the basement membrane becomes disformed, this abnormality, it is disrupted by the help of some enzymes like collagenase and hydrolysis, or the metalloproteinase enzymes that into collagenase. You know the basement membrane is made of collagen, and collagenase enzyme disrupt the membrane and it will invade it into different parts of organ and also in the blood vessels. And also damage the cell-serine contact. It is lost by the inactivation of ecadherins. Ecadherins maintain the cell contact with each other, make them together, but when the ecadherins inactive, the cell will spread and invade it to different sites. And then it will be stage of metastasis, in which it will spread to distant organ. After going to blood vessel and lymphatics, it will spread to other organs, for example, liver, lungs, heart, brain, etc. So spread to distant organ where the lymphatics in. Like if we are talking about the ovaries, in ovaries you will get nodule with the menstrual cycle happen, and then the retrograde blood, the retrograde flow of the blood help to metastasize this cancer to different organs like lungs, liver, heart, and the brain.